another speaker, that is Kate Adamala. Thanks. So Kate is a biochemist that built synthetic cells. She began her studies at the University of Warsaw, and then she graduated from Harvard, where she worked with Nobel laureate Czech Shostak. I can hear you, yes.
Can you wave if you hear me?
Yes, we can.
Okay, so you can hear me, I cannot hear you.
All right.
I was just hearing Niels, so my headphones are correctly connected, and something changed when I became a presenter.
I hope others can hear me, Justina.
In the interest of time, unless there's something you have to tell me, that we can just move on.
No, we can move on.
All right, so let's just move on, and I will not hear you. This is bizarre.
Can you see that?
I was just hearing everything that Niels was talking about, and now I can't. Okay, never mind. As long as you hear me, I will share my screen and
1, 2, 3.
And move on. All right, so can I share my screen? You need to wave to me because I don't hear you. All right.
Go ahead.
Okay. So thank you very much for having me here. And I'm very, very, very happy to be able to talk at this first Polish Astrobiology conference. And what I'll talk about today is how to investigate the origin of life, starting from something that's absolutely not living and really, really simple. And that is reconstituting the LUCA, so our last universal common ancestor, using simple biochemical experiments. So this is kind of a diagram that's probably very familiar to all of you. This is what had to happen on a prebiotic Earth. And we started with molecules, and then the molecules started self-organizing until out came life. And how do we investigate that? How do we investigate those processes? And the way we have right now is basically just two different techniques. We can either investigate in vitro processes, or we can investigate whole life cells, as we just heard in the previous talk. And there's this kind of a hole in the middle, because if you work with simple compounds, it's relatively simple, but it's not really biologically relevant. And on the other hand, if you investigate life cells, you don't really know what you're working with, and they're way too complex to even approach the last universal common ancestor. So this is the field that I'm in, the engineering synthetic cells. So we're engineering those really simple cell-like entities that are not really living to try to look at how the last universal common ancestor could have looked like. And this is the assumption we're making, that those simple cells mimic the processes that could have happened during the origin and the earliest evolution of life. And there are two kind of different ways of making those simplest cells, either making complex natural cells simpler, or building from the top, kind of a bottom-up approach, where we take simple compounds, put them back together to make something that looks like a cell. And so to me, a synthetic cell is everything that did not come from the last universal common ancestor. So any lab-like entity that we made in the process of those experiments. And the synthetic cell experiments are more complex than biochemical experiments. So there is some emergent properties in the system, but they're still fully understandable and fully controllable. And there are things you can control in synthetic cells that you cannot really control in natural cell systems. And that includes things as fundamental as size of the cell, composition of the cellular membrane, but also the type of translation system, the type of the DNA genes we're using in there, or even alternative genetic systems other than the DNA. And when I say synthetic cell in my lab, I understand it as a liposomal bioreactor that's capable of expressing proteins. So it's essentially a central dogma, modified central dogma, encapsulated inside a liposome. And a membrane is just a regular phospholipid membrane, like your own natural cells. Sometimes we don't use membrane at all. We just use droplet microfluidic systems, where we have those little droplets that all look very similar or similar in size don't have a membrane but do contain the central dome, basically the artificial cytoplasm. And the interesting stuff is what goes inside, and that's the sulfate protein expression system. That's the cytoplasm of synthetic cells. And that cytoplasm gets made either by purifying natural translation systems, like in this case from E. coli, or by putting together biochemical reactions entirely from scratch. What we do in most of the experiments in my lab, it's purifying the E. coli ribosomes. So what we're doing is we are essentially a chemistry lab that pretends to do biology. We're putting together cell-like systems, one molecule at a time, trying to reconstitute those earliest lifelike behaviors of the last universal common ancestor. And so here's an example of one of the projects that we did, and I'm going to show you two examples just to show you the boundary capacities of this system. So in this case, we have synthetic cells that express a protein inside and export it outside. Sounds simple, all cells do that, right? Except this system is stone dead. These are non-living synthetic cells. So we express a protein inside the cell, and then we tag that protein with a tag that allows that protein to wiggle out of the membrane. And just to give you an example of what kind of techniques we use to analyze the experiments in my lab, what we do is we label the membrane in red, and we run a so-called size exclusion column, where we purify the liposomes, the synthetic cell liposomes, on a Sepharose column, where we separate big things from little things, essentially. And as you see, the peak is in a red fluorescent channel that corresponds to the membrane. So this is where the synthetic cells are. And the same sample can be imaged in a green fluorescent channel. And you see that in the sample with the cell-penetrating peptide tag, most of the protein is outside of the synthetic cells. So the peak of the sharp red peak that signifies cells does not overlap with the big green peak. So all protein, most of the protein we made is now outside. The control without cell-penetrating peptide tag shows that the cells remain intact, but all the protein is still inside. So what can we do with it? One of the things we can do with this system is drag a payload across the membrane. And so that's what we do, is we drag a RNA payload across the membrane. We fuse an RNA binding protein with a fluorescent RNA aptamer, and we drag that across the membrane. Another thing we can do is something useful, and that is closing a functioning genetic circuit. In this case, it is two populations of synthetic cells. One of them expresses T7 RNA polymerase. The other one has a T7 controlled GFP, green fluorescent protein gene. For this to work, the one population has to make the T7 RNA polymerase and export it outside. And then the other population will make the GFP only in presence of T7. So what happens here is if we mix the T7 and GFP populations at equal amounts, we see GFP fluorescence. So the T7 RNA polymerase is made in one population, is exported outside, and then is imported into another population of synthetic cells. If we double the amount of the T7 population, we double the signal we get from the GFP population. If we double the GFP population, we get no increase in signal, which indicates that the rate-limiting step in this reaction is the expression and export of the T7 RNA polymerase. And all the necessary negative controls show background fluorescence. So that shows you this works. That shows that we can export genes, we can export payloads across a membrane of a synthetic cell. We're developing the system further now to investigate things like horizontal gene transfer. And another example of the synthetic cell system that behaves very much like a possible simplest living cell is ability to acquire antibiotic resistance. So what we do in those kinds of experiments is we have two populations of synthetic cells. One of them has resistance to antibiotic, in this case kanamycin. The other one does not have resistance to that antibiotic. And both of those populations have green fluorescent protein as an indicator of the health of the synthetic cell metabolism. And when we mix each of those populations with antibiotic, in presence of antibiotic resistance, we see green fluorescence from the metabolism of a synthetic cell that's resistant, and we don't see any fluorescence in the ones that are not resistant. Now, if you look at, I'm missing one picture apparently on this slide. If you look at the red channel, you can see that the membrane integrity is maintained. We can also quantify the fluorescence, and you can see that as we increase the concentration of kanamycin in the samples with no antibiotic resistance, the fluorescence of which indicates health of the metabolism goes down significantly, drops to 0 to undetectable levels at higher concentrations, and in presence of resistance, there's some metabolic cost to pay, but not much. So the fluorescence is still detectable. What we can do now is we can prove that the membrane channels are what is actually necessary to bring the antibiotic into the cell. And so we have two populations. One has a red fluorescent protein called mCherry as an indicator of metabolism. The other one has GFP. The one that has GFP has no membrane channels. So the assumption is only the one that has membrane channels, in this case alpha-chemolysin membrane channels, only that population can uptake antibiotic. And sure enough, the population that has metabolism labeled with GFP does not indicate any stress in presence of antibiotic. The population that has channels, the health of the metabolism, here shown as the expression of the red fluorescent protein, goes significantly down. Now what we can do is we can investigate transfer of that antibiotic resistance. So we have two different populations of synthetic cells. We fuse them or mate them together. And the way we do that mating is we label the membranes with complementary DNA tags. And those DNA tags make the membranes to fuse, just like the snare proteins make your synaptic vesicles fuse with your synapse. So we have those two populations. One has GFP labeled metabolism, the other one has channels. So if you remember from our previous couple slides, in absence of channels, the metabolism should be healthy. So in the absence of fusion, the metabolism stays healthy in presence of kanamycin. But if we do induce fusion or mating, the daughter population is susceptible now to antibiotic. So the fusion or mating in this case is harmful to our synthetic primitive cells. Now, if we have two populations again, and one of them has antibiotic resistance, and the other one has GFP labeled metabolism and channels, if we fuse them, in the absence of fusion, the green labeled metabolism goes down because antibiotic can go in. But if we fuse them, the daughter population has kanamycin resistance now. And so the fusion in this case is beneficial to cells, and it demonstrates the transfer of antibiotic resistance. So just summarizing this, those experiments show that you can have those synthetic, very primitive cells behave like lifelike norm cell populations, in that they have antibiotic resistance, they can transfer that resistance, they can have not just resistance to antibiotic, but they can also mate, and their daughter populations can have the resistance. So we're working on developing that system to get kind of a more complex behaviors out of it. So this is kind of a summary slide that shows you that synthetic cells are, hopefully shows you synthetic cells are awesome. You can do a lot of different applications, even beyond astrobiology. A lot of the things that the field is working on are very practical biomedical applications. And speaking of those biomedical applications, and to tie that into the astrobiology theme of this conference, I want to show two things. One is, a lot of the time people talk about how are we going to look for life in the universe, and especially in the solar system, but we don't really know what we're looking for, because we don't know what kind of life can we find on Enceladus or on Europa or even on Mars, or even if there's a shadow biosphere on our own Earth, because we don't know it, because we only have one type of life right now. We've only studied one type of life, and that's where synthetic cells can be really helpful, not just in recreating the last universal common ancestor of life on Earth, but also on showing alternative pathways to evolution, what other life-like systems could look like. And another application of synthetic cells is in enabling long-term space exploration. And so I was lucky to be part of this amazing team that set out to set first synthetic cell experiments into the low orbit on a sounding rocket to see if systems like that can even survive under spaceflight conditions. So what we did is we had two different experiments. One is a synthesis of a functional RNA aptamer, fluorescent RNA aptamer, and the other one is a protein expression, simple protein expression in synthetic cell system. So we lyophilized the protein and the transcription systems. We packed it up on a CubeSat. We launched it. I'm showing that video because to me that was super exciting because that was the first time my own payload went up on a rocket. The rocket didn't explode. It was a sample return mission. So we got the samples back. We got to analyze it. And now we can say synthetic cell experiments can actually survive under spaceflight conditions. And so the next step would be to prepare experiments for actual human astronauts to perform on the ISS. And those experiments will be using synthetic cells to control life cell bioreactors. So this is a kind of a similar to what Niels was doing, except we're controlling those bioreactors with a layer of synthetic cells. So we're trying to control a synthetic cell with a physicochemical trigger, a small molecule or light, and then the synthetic cell in turn can control activity of a life cell bioreactor. Hopefully we'll have data from that project to report in a few months. So last but not least, there's literally dozens of us. The synthetic cell community is growing. We are a relatively new field. It's been only about a decade or so when people can with a straight face say I will make a synthetic life and it's not considered crazy. And we are a very dynamic, very friendly community that's aiming at trying to build cell-like systems and then have those cell-like systems behave like natural cells, investigate all the properties of natural cells, but also build orthogonal living systems for studying the origin of life, for studying exploration, space exploration, enabling long-term human space flights, and also looking at building life as it could look like on other planets in the solar system. And that I will conclude my time. Hopefully I made up enough time to give you some of your break back. I want to thank all of the funding agencies. I want to thank everyone who works in my lab, and I want to thank all of you for having me here. I will.
Thank you, Kate. I know you can't hear me,
but I see Tomek is waving.
Yeah, so
and I'm super frustrated that
we have some questions.
I do not hear you.
I know.
It is annoying. Yes, I can type. Let's see. Oh, I see Q&As. Okay, I can start answering those questions from the Q&A. Okay. So, question. How antibiotics act on such synthetic cell? So in this case, the antibiotic is kanamycin, which is a antibiotic that blocks ribosomal translation. So the antibiotic can act whether the system is alive or not. And in our case, it's a stone dead system, but antibiotic can still kill it further, which means it can induce a, basically it can stall the ribosomes, and that's how the antibiotic works in our case. What is the stress that antibiotic causes on synthetic cells, and how do you measure it? The stress is measured as a function of the protein production. So we're looking at the production of the protein in our synthetic cell system, and that production is proxied by green fluorescent protein, because that's a signal we can measure. And so in presence of antibiotic, the ribosomes stall, and we have no GFP. In absence of antibiotic, everything moves smoothly. Next question. How does the import of proteins work? Are the membranes so permeable for proteins? If yes, how do the proteins that you need inside stay inside? So the membrane is not permeable to anything. It's not permeable to even small ions. It's a very impermeable phospholipid bilayer membrane. The way we export proteins is using those cell-penetrating peptide tags. The one we're using in those particular experiments is derived from a protein called penetratin, which is a natural cell-penetrating peptide protein. There was this big hype in the field long before I started my academic career, where people thought cell-penetrating peptide tags will be the solution for drug delivery of therapeutic proteins to cells. It didn't work out in live organisms, but it turned out that there is a lot of those proteins that can shuttle payloads, in this case small proteins, across a phospholipid membrane. And the way this cell-penetrating peptide tag works is it's very simple. It's essentially a hydrophobic tag that wiggles through the membrane. And in this process of wiggling, it carries a payload. There are limits to it, obviously. For example, we cannot carry long RNA payloads. The reason I only showed you carrying a fluorescent RNA tag payload is because we failed at carrying longer payloads. We tried to carry mRNA payload, and that didn't work. So that's the limit of that small cell penetrating peptide tag. And then another question: catalysts are often used in creating. Oh wait, that's a question from the previous talk. Okay, so I think I managed to answer all the questions, and because I don't hear you, no one could stop me. This is dream talk, basically.
Yes. So thank you
I will say
everyone for
thank you very
listening.
much.
Thank you, Kate, and have a break. We're having a break now, and we meet again for Sarah's talk.